All right, good morning, everyone. Welcome to the CIC lecture, the last one for April of this year. So I'm very excited to introduce Chloe Savignac. I've seen some of her work. I had some sneak previews because I'm sitting on her committee. So I think this is great timing for her to present, the massive amounts of effort she put into her master's work so far. She's a second-year master's student, in McGill's integrated program in neuroscience, and she's supervised by Dr. Danila Stock. And prior to joining Dr. Stock's lab, Chloe graduated with first-class honors with an undergrad in cognitive neuroscience at McGill. Her current research efforts are focused on applying tools from big data analytics to answer long-standing neuroscientific questions in large population data sets. As part of her MSc, Chloe is investigating covariation between the hippocampus and default node network in populations of healthy individuals at risk for Alzheimer's disease. So I'm excited for your talk, Chloe, and thanks so much for taking the invite. I think there's probably more people than you see here actually attending, because I think a lot of my lab likes to come together to watch it in little pods. So they're communing over your talk. So with that, I'll let you take it away. Thanks so much.
Perfect. Thank you, Malar, for the introduction. So as was mentioned, I'm a second-year master's student at McGill's integrated program in neuroscience, supervised by Danila Stock. And today I will present the results from the first manuscript of my master's, which is titled APOE2 versus E4 dosage, shows sex-specific links to hippocampus-default network subregions covariation. So epidemiological studies have shown that the APOE gene plays an important role in late onset Alzheimer's disease. So the APOE allele is determined by two single nucleotide polymorphism, or SNPs, that I'm showing here on this slide. So a mutation on this first SNP causes the E4 allele, which is associated with increased risk of developing late onset Alzheimer's disease. A mutation on this second SNP is associated with the E2 allele, which is known to decrease the risk of developing late onset Alzheimer's disease. So the majority of clinical studies have focused on the E4 allele, so the bad genotype. And although it was mostly investigated in terms of carrier versus non-carrier status, so as a binary classification, there is evidence of a dosage effect in the risk conferred by APOE E4. So each additional E4 allele could increase the odds of developing Alzheimer's by 600%. There is also evidence of sex specificity in this dosage effect. So in female, it is thought that a single E4 allele is sufficient to increase the odds of developing Alzheimer's disease to the same level as in E4 homozygous males. So in contrast, carrying an E2 allele has been associated with reduced rate of amyloid beta accumulation and cognitive decline. And each additional E2 allele could reduce the odds of developing Alzheimer's by 60%. So clinical evidence suggests that E2 could still be protective against cognitive decline in the presence of an E4 allele, which makes its protective effect on the brain a promising therapeutic avenue. Nonetheless, the rare prevalence of E2 homozygote in the general population makes its effect on the brain almost impossible to investigate in traditional neuroimaging samples. So over the past 2 decades, brain imaging studies have converged on the disruption of a network of higher association regions that involve key nodes of the default network in individuals with Alzheimer's disease compared to healthy control. Most biomarker studies have attempted to identify functional and metabolic patterns that reliably tell apart E4 carriers from non-carrier. Yet, most other APOE variants have been largely neglected, perhaps because they occur much more infrequently in the general population. In addition to disruption of the default network, changes in the microstructure of the fornix, which is the main output tract from the hippocampus to the association cortices, have been reported in individuals at risk of Alzheimer's disease before the onset of clinical symptoms. Damage to the fornix was subsequently identified as an accurate predictor from progression from mild cognitive impairments to dementia, and could potentially represent a link by which subcortical region of the hippocampus and neocortical regions of the default network are together affected in Alzheimer's disease. So as I just mentioned, the hippocampus has direct anatomical connection to the default network through the fornix, and is also thought to be affected in early stages of Alzheimer's disease. Among the few studies that could investigate concurrent connectivity alteration in the hippocampus and region of the default network in E4 carriers, the hippocampus was typically treated as a monolithic structure. rather than appreciated its functional and structural heterogeneity. Indeed, we know from early histological staging from the late 90s that there are subregion-specific alteration in the hippocampus in early Alzheimer's disease progression, especially in the CA1 regions during Braak stages 2 and 3. Nonetheless, current neuroimaging study continue to consider the hippocampus as a single node when investigating its coupling with default network regions or when looking at atrophy over the course of Alzheimer's disease development. The recent avenue of ultra-high resolution atlases and advanced automatic subsegmentation techniques now allows for the assessment of the subfield-specific vulnerability of both hippocampi to Alzheimer's disease progression in healthy individuals before the onset of clinical symptoms. So instead of relying mostly on postmortem autopsy from patient to ultimately confirm Alzheimer's disease diagnostic, we will soon be able to directly, non-invasively, quantify the level of risk of a given patient based on subfield level granular information. So these new automatic subsegmentation techniques allow to move away from small clinical samples to population data sets, which in turn allows us to more accurately describe the structural correlates related to Alzheimer's disease risk in the general population. So this is precisely what we're aiming to do here. So by capitalizing on the rich phenotyping available for 40,000 UK Biobank participants, our study could confront the effect of APOE E2 and E4 on inter-individual expression of hippocampus-default network covariation, which is something that was out of reach in traditional small to medium brain imaging samples. We benefited from subregion level granularity to describe Alzheimer's disease-related patterns of deviation in neocortical regions of the default network in lockstep with subregion of the hippocampus in a multivariate approach. We are thus in position to zoom at a microstructural level previously reserved to postmortem investigation, but that is now scalable to population dataset. By working with this broad prospective cohort, we are also especially interested in looking at a broader range of phenotypes that the UK Biobank offers that are not usually available in traditional neuroimaging data, but that have been identified by the Lancet Commission as being related to Alzheimer's disease risk, such as socioeconomic determinants, lifestyle factors, environmental quality, and social network. Working at the population level also allows us to assess the impact of less prevalent genotypes, such as E2 homozygotes. And in doing so, we're uniquely positioned to eliminate possible sex association between rare APOE gene variants that previous brain imaging investigation systematically ignore. So the first step in our analysis workflow consists in doing a two-pronged decomposition of subregion level covariation between a set of 38 hippocampus subregions and a set of 91 default network subregion. So to do so, we use canonical correlation analysis, which is a doubly multivariate statistical technique that is looking for a latent pattern of covariation between two sets of variables, which here are the sets of hippocampus and default network subregions. And we use T1 weighted MRI data from around 40,000 UK Biobank participants. Once we extracted these different modes of hippocampus default network covariation, we wanted to extract structural deviation in these different structures as a function of dementia risk. So we perform a bootstrap different test of the CCA solution with regards to family history of Alzheimer's disease and related dementia. Next, we perform a deep phenotyping of hippocampus default network covariation expression, which consisted in a phenome-wide profiling across 1,000 UK Biobank traits that span 11 broad risk categories. Lastly, we tested for association between established risk factors for Alzheimer's disease, APOE genotypes, and hippocampus default network covariation pattern to really look at these rare genotype that previous neuroimaging investigation were not in position to explore. So here a schematic of our analysis workflow. So as I said, the first step is to perform this canonical correlation analysis. So we have a first set, I'm like using my mouse to show you. So we have this first set of default network subregion volumes for our array of 40,000 UK Biobank participants. And we have another set of 38 hippocampus subregions volume for the same 40,000 UK Biobank participants. So what the canonical correlation analysis does, it finds the linear combination of default network subregions volumes that makes them maximally correlated with a linear combination of hippocampus default network subregions volumes. So we're really looking to maximize the correlation between the two sets. And once we find the first set of linear combination, we extract the variance accounted for by this first combination from the data, and we find the second set of linear combinations that account for most of the remaining variance. And we can do so iteratively for a number of time that is equal to the length of the smaller set, which here is 38, the number of hippocampus subregions. So we decided to focus on the first 25 modes of hippocampus default network covariation. And by construction, these modes represent distinct, uncorrelated, and hierarchically ordered patterns of covariation between the two sets of hippocampus and default network subregion. So distinct because once we find one mode, we take the variance accounting for by that mode out of the data, and we find the second mode that accounts for most of the remaining variance. So by construction, they are uncorrelated. And they are also hierarchically ordered in the sense that mode 1s account for more variance in the data than mode 2, which in turn accounts for more variance in the data than mode 3. So CCA is not usually used to reveal group difference at the single variable level. As I said, it's more of a latent method. So it's used to build, to derive this kind of latent mode of covariation. So we had to build an analytical framework, especially designed to extend inference from this latent dimension to subregion specific information, which were really the input of the CCA analysis. So to do so, we use a bootstrap different test, and we built a non-parametric distribution of group contrast estimate. So over 100 iteration, we shifted participant sample a little bit and recomputed the CCA solution. And in participant with and without family history of Alzheimer's disease. And we stored the weight associated with each subregion on each of the 25 mode. So this is what I tried to illustrate here. So let's say we have this first region here, the dorsal-medial prefrontal cortex. For mode 1, we, over the course of these 100 bootstrap iteration, we have computed the weight associated with these subregions in participant with family history of Alzheimer's disease and related dementia, which we see here in this red distribution. And we have done the same for participant without family history of Alzheimer's disease, which we see here in this blue distribution. And we looked for the difference between these two distributions, so the weights in participant with and without family history of Alzheimer's disease. And we considered statistically relevant brain regions that had no null effect within a two-sided 80% confidence interval. So this is what I tried to illustrate here. So we kept the regions that had statistically relevant deviation. So here, it's a good time to take questions for this part of the analysis, because this is really the backbone of the rest of the presentation. So if I can answer a question right now on this analysis workflow, it will be a good time. If, oh, yes, I see.
So it's worth reading it out loud, Chloe?
Yeah. So the question is, the 38 subregions are derived from ex vivo data, correct? What is the resolution of your in vivo image? And is this fine-grained enough to derive a reasonable volume estimate of the 38 regions? So that's a good question. So the atlas we use is a combination of both ex vivo and in vivo data. And the resolution, I had it, I think I mentioned it on this slide, or oops, sorry, on the other slide. So it's about 0.001 millimeter cube. So it is fairly reasonable. It is part of the, I will go back to my slide. It is part of the FreeSurfer 7.0 suite. So it is based on a probabilistic atlas that also takes surrounding region of the hippocampus. So it has been widely used in the UK Biobank. And we do have some fine-grained microstructural information. So they have segmented in terms of the different groups of neurons, right? So there are different layers that they were able to identify. with like pyramidal cells and the different CA fields. So I can go more in depth at the end of the presentation if that's of interest. But yes, this atlas has been widely used as well. So as a target phenotype, we use self-reported family of Alzheimer's disease as a single but accurately measurable estimate of Alzheimer's disease risk. So genome-wide association studies have revealed that when dealing with late onset diseases, such as Alzheimer's disease and related dementia, using proxy cases, that is, the relative of affected individual, could allow for more complete characterization of disease risk amongst individuals before the onset of clinical symptoms. So in particular, self-report of family history of Alzheimer's disease and related dementia in the UK Biobank, which is precisely the same phenotype at the core of the present investigation, was found to replicate established risk loci from case control investigation, as well as to identify novel loci in healthy participants of the UK Biobank. Working with proxy cases also allows us to boost the sample size and thus the statistical power for quantitative analysis to identify more suitable effect related to these rare genotypes, such as E2 and E4 homozygotes. So of our sample of around 40,000 UK Biobank participants, around a quarter of them had family history of Alzheimer's disease, and the majority was on their mother's side, and minority on their father's side, and then a few individuals had both maternal and paternal history. It was fairly evenly distributed in males and females and a mean age of 55 years old. So we see that the distribution of APOE genotypes is really similar to what we found in population of European ancestry. So we have these low prevalent genotypes such as E2E2 with a prevalence of 1%. We have E2E4 with a prevalence of 2% and E4E4 with also a prevalence of 2%. So we first conducted a phenome-wide analysis to systematize domains of UK Biobank traits in terms of their association with hippocampus-default network signature and Alzheimer's disease risk. So as I previously mentioned, we are especially interested in looking at a broader range of phenotypes that the UK Biobank offers that are not traditionally available in neuroimaging investigation. So I will show the top 3 modes of hippocampus-default network covariation that had the most brain behavior associations. So first here, what I'm showing are the subregions that had statistically relevant deviation in relation to family history of Alzheimer's disease. So what we see here on the left are what we call hits, so subregions with significant statistically relevant deviation in the hippocampus. So we see really strong weight in the CA1, CA2-3 and molecular layer. So as I mentioned, the molecular layer is where the axon of the pyramidal cells from the CA fields are located. And we see these strong weight on the left side, as well as on the right side for the molecular layer, the CA2-3, dentate gyrus, CA4, as well as the hippocampus tail. And then if we look at the patterns of structural divergence on the default network side, we see strong hits in the anterior midline structures, such as the default medial prefrontal cortex, as well as some posterior midline structure, the precuneus retrospinal cortex, posterior cingulate cortex, and some lateral hits as well. So ventrolateral prefrontal cortex, inferior parietal lobule, and superior temporal gyrus. So next, we wanted to know how is the individual expression of this mode with these specific subregions that differ in individual with and without family history of Alzheimer's disease is related to a broad range of phenotypes in the UK Biobank, given the genetic risk of each individual to Alzheimer's disease. So to investigate that, we created a bivariate dosage scale to test for the opposing effect of APOE2 and APOE4 on ADRD-related phenotypes. So this scale ranged from -2, which meant 2 E4 allele, to +2, which meant 2 E2 allele. And using this scale, we fitted linear regression models to explain the variance in this bivariate dosage scale as a function of inter-individual expression of mode 1 in this case, but we did it for all of the 25 modes. And we conducted these analyses in males and females separately to really highlight sex-specific effect. So once we fitted these linear model, we were able to predict the APOE dosage that is associated with each subject-specific expression of mode 1. So putting it more simply, we asked, what would be the expected E2 versus E4 dosage of this specific participant, giving their individual expression of here mode 1. So this gave us a score between -2 to +2, which represented the genetic risk associated with a subject-specific expression of, in this case, mode 1. So then we took this genetic risk that was pooled across individual expression of mode 1, and we correlated to 977 UK biobank phenotypes, which is what I'm showing here on this Miami plot. So each dot represents a correlation between this predicted score that is pulled across individual expression of mode 1 and a set of UK biobank phenotypes. And the y-axis represents the negative decimal logarithm of the p-value. So the higher the dot, the stronger its p-value. And we have the correlation for males at the top and for females at the bottom. So what we can see here is a lot of environmental related phenotypes in males. So all these orange dots are related to air quality, NO2 air pollution, there are green space, natural environment, on location, so really related to the quality of the environment you're in. And we also see a preponderance of these cognitive hits in blue. So these are all cognitive traits related to verbal numerical reasoning, which were part of the fluid intelligence battery of the UK Biobank. And for males, we also have some lifestyle hits, such as time spent watching television, getting up in the morning. And then if we look at females, we have some cognitive hits as well, but to a lesser extent, and the p-value are less robust than in males. But we have these sets of like cardiovascular related factors, such as insulin growth factor 1, hematocrit percentage, ventricular rate, and paternal history of heart disease. Then I repeated the same, as I said, I did the same analysis for all of the mode. Here I'm showing the top 3 modes with the most brain behavior association. So in this case, this is mode 3. So mode 3, we see that on the hippocampus side, the structural divergence are really more focalized, right? We see that it's in the fimbria and the presubiculum. So the fimbria is where the axon from CA1 and the subiculum merge to form the fornix, which, as I said, is the main output tract of the hippocampus to higher association cortices. So we see that in this mode, the divergence that are related to family history of Alzheimer's disease are focalized to the fimbria and presubiculum. But then on the default network side, we see an overwhelming structural divergence across the whole default network. So all of the regions are in yellow, so there's strong deviation that are really focalized to the fornix on the hippocampus side, but then related to the whole default network. So I did the same process and I pulled genetic risk across individual expression of mode 3. And this is the association we obtained. So we have, again, a high preponderance of environmental phenotypes. So NO2 exposure, green space, particulate matter, which is fine particle of, sometimes it's from cars, so emissions that are present in the atmosphere, natural environment, and then cognitive hits in males. So in females, we have some environmental factors as well, but to a lesser extent than in males and with smaller p-values, so a less robust association. Same for cognitive hits. So here we see them in males, they cross the Bern-Ferroni multiple comparison threshold, but they do not in females. So these cognitive hits here do not cross the Bern-Ferroni threshold in female. Then here, mode 8 showed the third highest number of brain behavior associations. So if we look at the structural divergence, we see that it's really focalized to the fimbria again. So even though we use a multivariate procedure and we looked at 38 hippocampus subregions, we have this really specific localized effect on the fimbria with no accompanying divergence on the default network. So this mode really shows Alzheimer's disease related divergence to the fimbria. So it's really the fimbria. And if we look at the sets of phenotypes that are associated with individual expression of this mode, we see that they're all related to body mass, height, and cardiovascular phenotypes. So these are all in gray, phenotypes related to weight. So we have water mass, fat-free mass, height, and also some early life factors such as birth weight, comparative height at age 10, and same in males, but we see that there are more of these BMI-related phenotypes in females. And again, we have the presence of cognitive hits in males, but not in females. So we next systematically explored nonlinear association between established ADRD risk factors and the 25 pairs of hippocampus-default network covariation patterns across the different APOE gene variants. So our goal was to probe for clusters of risk anatomy links that are most related to the derived patterns of hippocampus and default network covariation in these different APOE genotypes. So to this end, we used an hierarchical clustering approach that allowed us to assess the relative importance of ensuing clusters of risk anatomy links in each of the different APOE genotypes. So the first step for this analysis was to multiply the population-wide hippocampus and default network covariation pattern by each of the APOE genotypes coded as 0 or 1. So for each of these dendrograms here that I show, I multiplied the individual expression of each of the mode on the hippocampus and default network side by the genotype encoded as 0 or 1. So binary encoding, such that participant without a given genotype were zeroed out. So we obtained 6 new population-wide vector, 1 for each APOE allelic combination, and we did so to create a nonlinear term that we could then Spearman correlate to 63 pre-selected Alzheimer's disease risk factors that we have previously shown are related to Alzheimer's disease risk in the UK Biobank and Canadian longitudinal study on aging. So we then performed an agglomerative clustering analysis on the Spearman correlation coefficient, which consists in repeatedly merging coefficient with similar variants together until all observations are merged in a single cluster. So here what I'm showing are the dendrograms, which represent the distance between each cluster identified after the different level of branching. So the x-axis is the distance in the latent space where these clusters are formed. So I want to draw your attention on this robust cluster of social phenotypes. So we see it here for E2, E3 homozygous, family relationship satisfaction, friendship satisfaction, and happiness. There are here as well for E4, E4 homozygous at the top, and as well as for E3, E4 here. And they were also present in E3, E3 homozygous and E2, E4, although not shown here. So these 3 phenotypes represents, in general, how satisfied are you with your friendship and family relationship, which is a subjective appraisal of one's social bonds, which is also intimately tied with loneliness, right? If you feel like your social needs are not met or are inadequate, you will experience what we call subjective social isolation, so a feeling of loneliness. So build on this pattern, this social cluster that is present across most APOE genotypes, builds on previous research from our team, where we have shown that perceived and objective social isolation, such as loneliness and lack of social support, are constantly associated with classical Alzheimer's disease risk factors in the UK Biobank cohort. Next, if we zoom in on specific APOE variants, such as E2 homozygotes, we see the early branching of all of these personality traits related to neuroticism. So irritability, suffering from nerves, having fed up feeling, feeling miserable, and having mood swing. So these are all personality traits that are associated with neuroticism. So, and we see that it's an important personality trait because it branches really early in E2, E2 homozygote. In comparison, if we look for E4, E4 homozygote, we see that there's the early branching of age-related phenotypes, such as macular degeneration, angina, retirement, and age, which could say that these age-related phenotypes are more important or characteristic of E44 homozygotes.
Sorry, Chloe, I know you asked for questions later, but what are the numbers on the y-axis? Can you just repeat
yes. So on the y-axis, I retained 4 levels of branching. So we could have continued until all risk factors are a single node. So if there's a number on the y-axis, it means that this branch contains 4 different risk factors that are not yet kind of separated. So they're really close invariants. So I could have continued the branching until each risk factor is a single node, but I wanted to identify clusters. So I kept 4 levels of branching to really have these broad patterns, like here, all of the neuroticism relating personality traits in the same branch. But yes, so this represents that we could have continued the branching further.
Thank you.
Welcome. So we next directed our attention towards sex-specific interaction between hippocampus and default network covariation regimes and APOE genotype status that would explain inter-individual difference in Alzheimer's disease risk. So to this end, we tested whether hippocampus default network signature systematically interacted with specific APOE genotypes in explaining variation in a collection of the same collection of risk factors that was previously investigated in the clustering analysis. So for these analysis, we individually regress each of the risk factors on 14 main parameters. So the main effect of the canonical variant being investigated, the main effect of each of the APOE genotypes, age, as well as 6 interaction term for the interaction of the canonical variant being investigated and the APOE, each of the APOE genotypes. So each column here represents a single model. So in each model, I regressed a given risk factor. So in this case, being a full or part-time student on these 14 parameters. And then each time we look at only one canonical variant from the hippocampus or default network side. So these first 25 columns are the models for the hippocampal canonical variant. And these other sets of 25 models are the canonical variants for the default network side. And these 2 first panels are the coefficient in males, and these 2 last panels are the coefficients for females. So we perform a label shuffling permutation test to assess the robustness of our findings. So in colors here, I only show the coefficient that differ from their respective null distribution in 95% of the time. So this was done to distinguish robust effect from noise because, as I said, each column is a single linear regression model. So we really wanted to make sure what we had here was robust. So we see these really strong interaction between genotype E2, and a lot of the canonical variants in males and also in females in estimating being a full or part-time student, which could be a social phenotype, right? A social risk factors, because we have to keep in mind that these are older adults. So if they're a student, it means that they went back to school. So not only are they exercising their cognitive reserve, they are also going back to a social circle and seeing people. And we see that this effect is even stronger in males. And this could be because we know that as couple age, the female is often responsible for the social circle in older couples, and male tend to have these kind of more superficial relationship where they meet people in group activities, so like sports or clubs. And it's often the female that is responsible for maintaining these more long-term relationships. So probably being a full of part-time student kind of interacts more with the brain in males than in females, and it seems to be particularly affecting E2. Similarly, we saw similar patterns of interaction between E2 and the canonical variant in estimating doing unpaid or voluntary work, which is also a social phenotype because volunteering is often done in groups. So it's a social engagement, which is also reinstating back a social network. And we see that these coefficients are again stronger in males than in female, and it's selective through the E2 homozygotes. Which was unexpected based on the literature because we keep focusing on the risk genotype, right? E4, and all this is affected with these risk factors. But here we showed that the interaction of the canonical variant, so the hippocampus default network covariation expression, interacts with E2, in predicting variants on these different risk factors that are related to social lifestyle. Then we saw another patterns of interaction between E2 and the canonical variant, but this time for later canonical variants, so in like 21, 22, and 24, but that was stronger in females. And here we're estimating engaging in strenuous sports, which could be a modifiable cardiovascular risk factors, right? So how active you are. We know that in female following menopause, there are higher risk of neurovascular complications, so possibly engaging in sport has this kind of stronger effect in females. And the fact that it interacts specifically with E2 is consistent with the literature that have shown that even though E2 is protective against cognitive decline, it is also associated with precipitated risk of cardiovascular pathology. So the last interaction I wanted to show is for physical environmental score. So this is the only phenotype for which we saw strong interaction with E4 genotype. So here we're estimating physical environment score based on genes and individual expression of hippocampus-default network covariation. And we see that we have this interaction between E4 and the canonical variants for estimating these physical environment score, which is a composite measure of air quality, air emission, and proximity to waste disposal or industrial sites. And this specific pattern of interaction with E4 homozygous is consistent with the literature that has shown that E4 carriers are especially vulnerable to the detrimental effect of air pollution. So in highly polluted cities, such as Mexico City, E4 carrier displays precipitated sign of cognitive deficits and amyloid pathology that can be seen as early as in childhood and adolescence. So we did a similar analysis, but this time instead of regressing a risk factor, I regressed family history of Alzheimer's disease, so binary, yes, no, on the same set of 14 parameters. So the main effect of each of the 6 APOE genotypes, the main effect of the canonical variant being investigated, the 6 interaction terms between the canonical variant and the APOE genotypes, as well as age. So here we see these really strong gradient effect, mostly in females. So we have E2 that is associated with lesser risk of having family history of Alzheimer's disease, and this coefficient is quite strong. In contrast, we see that E4 is also associated with higher risk of Alzheimer's disease, but it's also quite strong. And we have this dosage effect because we can look at E2, the coefficient for E2 are stronger than for E2, which are in turn stronger than E3. While if we compare in males, we don't see such a clear difference between E2 and E3. And then we see that in female, E2 still looks protective in the presence of an E4 allele because we have this difference in coefficient between E2, and E3 that we don't really observe in male. So the protective effect of E2 and the E2 allele seems to be stronger in females. And if we look here, we see that even in our sample of around 20,000 males, the main protective effect of E2 is not statistically robust in males. So it did not pass our permutation test. So that's why this first row is all blank. So it looks as if the protective effect of E2 is stronger or female specific. Lastly, I think the main takeaway from this slide are these patterns of interaction between the canonical variant and the APOE genotypes that we observe here on the lower rows. So at the top, it's male, bottom, it's female. And if we zoom here for female on the default network side, we see that E2 is protective against family history of Alzheimer's disease, right, against the risk, which is something that we know. But then if you have a brain structure that is highly expressive of mode 9 on the default network side, you have twice the odds of having family history of Alzheimer's disease, which is what we observe here with this really strong coefficient for the interaction of E2 and mode 9 on the default network side. So it looks as if sex and brain structure kind of modulate the protectiveness and risk associated with E2 and E4. And this is what mode 9 on the default network side looks like, because I know you guys would be curious. So if we look here, we have strong rates on midline structures, the dorsal-medial prefrontal cortex, as well as the retrosplenial cortex. And we have some negative weights for the inferior parietal lobule and for the temporal regions on the left side. So the last analysis we did, we run PCA to extract dominant principle of brain behavior association or latent embeddings from our hippocampus default network population signature. So the first step was to compute the correlation between the 25 canonical variants from the hippocampus and default network side and the same set of Alzheimer's disease risk factors from the previous analysis. And we ran PCA on the risk by canonical variant matrix and retained the top three, the three first principal component. So what I'm showing here is the projection of each of the risk factors on the three main axes of brain behavior association found by the PCA. So here on the first axis, we see that social phenotypes that were identified in the clustering analysis are highly expressed. So this is why we have these really strong weight for happiness, friendship satisfaction, and family relationship satisfaction. We have other social phenotype as well that are expressed, such as attending religious group, attending adult education classes, and the number of people in the household you live in. Then if we look at the second axis of brain behavior association, we see that socioeconomic determinants are highly expressed, such as fluid intelligence, education score, age completed high school education, alcohol intake frequency, time spent watching TV, and average household income. Then on the third axis, we have these sets of personality traits related to neuroticism that have these strong weights. So the same one that were highlighted in the clustering analysis for E2 homozygote. So miserableness, fed up feeling, mood swing, being worried or anxious, etc. So this axis is highly expressive of neuroticism-related personality trait. Then I conducted this analysis in male and female separately, just to see if the types of risk factors that would be emphasized on each of the principal axes of brain behavior association would be different. And we see that for the two first axes, it's mainly the same set of risk factors. So the social phenotypes here and there, the socioeconomic determinant on the second axis. But then this neuroticism related set of risk factors is not as expressed on the third axis in females. So in males, it is present, highly present, but not in female. So in females, we rather observe these phenotypes that are related to cardiovascular metabolism, such as heart attack, angina, stroke, hypertension, and diabetes. So it could suggest that neuroticism is a personality trait that interact with hippocampus default network covariation patterns in a male-specific way. So our population neuroscience approach has directly compared the putatively protective and deleterious consequences associated with E2 and E4 respectively. So by capitalizing on APOE genotyping and structural brain scan from around 40,000 UK Biobank participants, we uncovered sex-specific association between rarely investigated APOE gene variants and microstructurally defined hippocampus and default network signatures that are hardly ever discerned in a human, in a prospective human cohort. So across our signature of hippocampus default network covariation, we have identified the CA1 molecular layer, the fimbria, and their neocortical projection targets as being relevant for Alzheimer's disease risk. Our results are in line with the preferential vulnerability of CA1 in Alzheimer's disease patients, as well as with the episodic memory deficit that are attributable to microstructural alteration of the fornix. This resilience towards cognitive decline that is generally observed amongst E2 carrier could arise from a relatively higher baseline APOE steady state levels in region included the hippocampus and frontal cortex as compared to E4 carriers and E3 homozygotes. So this isoform specific effect that are related to the APOE protein could be further enhanced by microglia-driven homeostatic response to amyloid beta accumulation. So it is known that older E2 carriers with amyloid pathology are less likely to be diagnosed with dementia than E3 homozygote of the same age, possibly because they are more biologically efficient at scavenging the peptide. The presence of an estrogen-dependent enhancer in the promoter region of the APOE gene could in turn explain why the protective effect of APOE2 and the risk effect of APOE4 are stronger amongst female. So as I mentioned, E2 is usually protective against cognitive decline, but it was also associated with precipitated risk of cardiovascular pathology. So engaging in physical activity could therefore be particularly beneficial to older female E2 carrier in counteracting the rising risk of neurovascular complication that results from the combined effect of having an APOE2 allele and a decreased level of estrogen following menopause. So bringing support for this claim, we have shown specific interaction between hippocampus-default network signature and APOE2E2 in explaining variation in physical activity, which was uniquely observed amongst female. So in older age, a decrease in social activity that is possibly related to unemployment and or retirement could increase feelings of loneliness and consequently escalate the risk of cognitive decline in dementia. So social disengagement has indeed been associated with higher incidence of cognitive declines amongst older adults. And in contrast, social activity has been linked to an up to 40% decrease in Alzheimer's disease risk. So volunteering and being a full or part-time student, which are two social engagement that have repeatedly been flagged in our analysis on E2 homozygotes, could possibly counteract the pathological stress response that is seen in lonely older adults. And as I previously mentioned, this effect could be amplified in males because we know that in general, males have more superficial relationship that depends on their significant other as they age. So possibly social engagement kind of reinstate the social circle that has been lost due to retirement and just older age. We have also seen that neuroticism seems to be specifically correlated to hippocampus-default network covariation in males. And neuroticism is also linked to loneliness because it is thought that neurotic individuals have a higher time maintaining friendships and relationship in general. And so this neuroticism can in turn causes loneliness and this kind of chronic stress response that we see in lonely individual, which makes sense given recent evidence that have shown that neurotic individuals are at higher risk of developing cognitive impairments and dementia. So possibly this social engagement that we have seen in male could counteract or balance this kind of neurotic personality traits as they both kind of act on the physiological stress response. Then we have also highlighted a lot of environmental phenotypes that emerge as being E4 specific and enhanced in males. So we know from epidemiological study that traffic-related air pollution and residents near major roadways are associated with decreased cognitive ability. And there is evidence that this effect is increased in E4 carriers because they are more prone to neural toxicity in general in the brain. So in our analysis on mode 1, we have tied these environmental phenotypes to blood markers such as hematocrit and erythrocyte, as well as these subregion specific deviation that are known for early vascular leakage in the aging brain. So on mode 1, we show 19 default networks hit the prefrontal cortex, which is a region in which vascular and perivascular white matter damage has been observed in humans and canine chronically exposed to high level of air pollutant. The prefrontal cortex, especially Brodmann area 9 and 10, are also a sites of early reduced parasite coverage in the Alzheimer's disease brain. So we have linked these regions that are known for prone aggregation of pollutants, early vascular leakage, to these environmental phenotypes, possibly extending on the role of vascular integrity in protecting the brain from environmental stressor. And a reason why this effect could be enhanced, could be more detrimental in males, is that across species, female are known to show enhanced immune response and protection against oxidative stress. So they could be more resilient towards pollution-induced impairment. Lastly, across a lot of our analysis, Social relationships have emerged as showing strong risk anatomy link across APOE genotypes. And this is in line with previous study by our team that have linked loneliness to midline structure of the default network in hippocampus, in which we have shown Alzheimer's disease-related divergence, such as the retrospinal cortex and dorsomedial prefrontal cortex, the CA1, and the molecular layer. So loneliness has previously been associated with a faster rate of cognitive decline, especially with its subcomponent of episodic memory that are sustained by the default network, such as immediate and delayed recall. So we know that friendship and social interactions are one of the most complex things that the human mind does. Yet, social isolation has only recently emerged as an important risk factor for Alzheimer's disease. Our analysis of risk anatomy link has especially uncovered a distinct set of social phenotypes that are related to happiness, family relationship satisfaction, and friendship satisfaction, and have emerged across most APOE gene variants in a way that were independent of E4 status and departed from traditional Alzheimer's disease risk factors. So quickly to conclude, while incumbent health policy has attempted to delay cognitive decline in vulnerable individuals, our findings rather spotlight modifiable risk factors that we have mainly linked to the protectiveness conferred by APOE2. We found especially strong ties between E2 and risk factor that could be exacerbated in the ongoing COVID-19 crisis, such as lacking physical activity and chronic social isolation. So the unprecedented scale and depth of our investigation into the E2 allele could open the window towards a new direction of effort directed at maximizing under-researched E2-mediated protective risk factors. So I want to thank my mentor, Danilo, as well as all the members from my team that have helped me in my analysis and in preparing this presentation. Thanks.
Thanks, Chloe. It's a tremendous amount of work. If there are questions, you can put up your hand or certainly feel free to put it in the chat and I'll read it out loud. So just the first question is from Cindy. She just missed the age range of the sample. If you could put up that.
So when participants were recruited, they were aged from 40 to 70 years old, and the mean age was around 55 years old.
Cindy, go ahead.
Hi, thank you for your presentation. I was wondering about the risk factors that you found. Could you talk a little bit more about what is like the main risk factors that you found more impactful?
Yes. So in terms, I think what was more striking was how these specific risk factors had the strong coefficient with E2. So this was, as I said, phenotypes that were related to social engagement. So being a full or part-time student and volunteering. And we saw that they had these specific patterns of interaction with E2. So this is more targeted. So if we look for a more kind of personalized medicine approach, these factors could possibly influence the protectiveness of E2. So if you have a patient that kind of is doing well, maybe these are things we can act on to make sure that we boost up the protectiveness of their genes by acting on their social network or their happiness in general. And there has been recent research that have shown that having a positive outlook on aging, such as a feeling of usefulness and just in general positive outlook on being older, amplifies the protectiveness of E2. So in participants that had a negative outcome on life in older age, E2 was no longer protective. So there was no difference compared to E4 carriers. So it seemed as if these things, either the quality of your relationship or just the outcome you have, can really act on the protectiveness associated with the genes. So I think these are important because they are modifiable risk factor we can act on. And then if we look at the more global perspective, so this is to have more of a bird eye view. So we did not zoom in on specific genotypes, but in this plot, we see that environmental pollution seems to have strong relationship with the brain structure and the risk, right? So this is also something that possibly we could act on, even though it's difficult to make a policy. But we know that global warming is one of the biggest crises we have right now, in addition to COVID. So I think these are two kind of modifiable risk factors on which we could act on, possibly, while the policymakers not us individually, but on which there is hope to act on.
Thank you.
I have some questions. You don't mind going back to that previous plot where you show the E2, E2 heat maps?
Yeah.
Yeah, so something like this, for example, right? So the allele frequency for E2 is about 8%, correct?
Just, you mean, well, E2 is about 1% and E2, E3, 12%, but yes, the E2 allele, I think it's around 7 or 8%, yes.
So E2, E2, so the prevalence of E2, E2 then in your sample is 400 people?
Yeah, around 400 people, yes.
And how many of those individuals were in school full or part-time?
That I could not tell you straight like this, but the percentage, but I could look it up after and tell you. But I can't remember on top of my head.
So are you worried that you're underpowered to come to the conclusion? Like when you break it down to cell size like this, are you concerned that you're underpowered to get to the conclusions that you're making?
That's why we did this permutation shuffling test. So I went a little bit fast on it, but so I randomly shuffled here being a full of part-time student, I randomly shuffled it across the whole participant cohort 1,000 times, and I recomputed the coefficient on these 1,000 iteration, and I built non-empirical distribution for each of the coefficient across these 1,000 iteration. So I think the permutation test kind of made the effect that were due to noise or by chance disappeared. So I'm fairly confident because of what we have here because of this permutation test that we did over 1,000 iteration.
Are you permuting across the whole sample or in the subsample of E2, E2 carriers?
Across the whole sample, but
how can you make that inference about the E2, E2 carriers if you're permuting against the whole sample?
Because it's there's a distribution for each of the coefficient, right? So there was this I made each time I recompute each of the coefficient. So I have like 1,000 coefficient for each of the different models, and I compare them to the real coefficient. So I think that test was fairly robust.
It'd be robust if you're saying something with the whole sample, or I worry that you're being too permissive, and you're generously estimating the p-value with that type of an approach.
Yeah, this is not a p-value because
or whatever.
Yeah, exactly. Yeah, we don't look at p-value usually because of the size of our sample. It just doesn't translate as well.
Yeah.
But it's close. Yeah, it's like closely similar. Yeah.
Okay. All right. Yeah, maybe something you want to take a look at because you know. So the other thing I have a question about is your interpretation. So, when I think of education and I think of elderly individuals or people middle age and older who are seeking education and new experiences, that in itself is indicative of a type of stimulation and probably a type of cognitive reserve, which I didn't hear you talk about at all. And so attributing to kind of the seeking out of education as a loneliness factor, sure, that's possible, but it also could be something completely different, right? And so it would have been interesting to hear your take on cognitive reserve in this context, or the building of cognitive reserve, especially at this older age. And also it'd be interesting to know how do factors that we know that are very much loaded on this variable, like SES, how are those related?
Yeah, so I think in terms of here, we're investigating the default network. So we know that one of the things that is mostly that activates the default network is really these kind of gauging social relationships. So when people are trying to compute relationship between others or having like a sense of their social network, we know that keynodes of the default network are activated. And recent research,
right? Sort of cognitive load. I mean, you could apply that very generally, right? Like
yeah, but in terms of how it's linked to the hippocampus, there has been recent development that have kind of extended this concept of cognitive maps to region of the default networks when we kind of navigate social situations. So this is we know that the hippocampus is really strong with the place set in terms of navigation. So this interpretation of why these specific subregions of the hippocampus and default network are together implicated, we link it more to these kind of social navigation activities, but it's true that it could be a cognitive reserve as well. That's why I think I briefly mentioned it. So it could be attributable to cognitive reserve, but something that links this plot and this plan together, which is volunteering, would be the social aspect, because in volunteering, there is maybe less of a cognitive kind of activity. So what could make link these different patterns of interaction together would be this kind of social factor that is closely tied to the default network function.
Okay. Great. I think we're out of time, but thank you so much, Chloe. Very interesting talk.